Hello, Professor Zurawski. As an addition to writing out the objectives for extra studying, I'm going to say them out for my benefit, so I figured might as well post it. Objective 1, Unit 1. Invertebrate versus vertebrate. Invertebrates animals have backbones, usually smaller, 95% of all animal species. Vertebrates, animals with backbones, usually larger, 5% of animal species, fish, amphibians, reptiles, birds, and mammals. Number 2, taxonomy, formal system of naming and classifying. Systematics, broader science of classifying organisms used to be based on visual and summary in biogeography. 4, ID the 3 goals of systematic zoology. Discover all species of animals. To reconstruct their evolutionary relationships, classifying animals according to their evolutionary relationships. 5. ID 3 domains of life, kingdom in that domain, and some examples. Archaea, bacteria, and eukarya. Eukarya kingdom is Archozoa, Euglensioa, Aviolata, Straminophilia, Rhodioplata, fungi, plantae, and animalia. List the 8 taxonomic categories, largest to smallest. Domain, kingdom, phylum, class, order, family, genus, species. 7. Use binomial nomenclature to properly name and ID an organism. Example, Daphnia coronata equal waterfly. 8. ID the difference between biodiversity, identified biodiversity, and estimated biodiversity. Biodiversity is the total number of species within an ecosystem and the resulting complexity of interactions among them. Estimated biodiversity is every year there's 7,000 to 10,000 species are named. Approximately 1.5 million species identified. Could be up to 7 to 10 million, some scientists think. There's less than 20% of all extant animals still in existence. Identified biodiversity is 5% all are prokaryotes and protists, 22% are fungi and plants, and the rest are animals. 9. Construct a probable evolutionary tree of life. Be able to construct a cladogram with an outgroup, 4 characteristics, be able to different stages, ancestral and derived, and be able to score the characters. Metric conversions. Bases are meter, liter, gram, and temperature. From highest to smallest, it's kilo, hecto, deca, base, deci, centi, milli. 12. The water temperatures. Boiling water is at 100 degrees Celsius. Freezing is at 0 degrees Celsius. Fahrenheit is 5 divided by 9, parentheses, the degree Fahrenheit minus 32. And then Fahrenheit to Celsius is 9/5 times the degree of Celsius plus 32. 13. Calculate density, area, and volume. Area is x squared. Density is mass over volume. Volume is 1 cubic meter squared, or 1 milliliter. 14. Measure a pipette, graduated cylinder, and note the meniscus. ID 15. ID the parts of a compound microscope in a dissecting microscope and be able to calculate the magnification. Dissecting. 16. ID the kinds of symmetry and their characteristics and provide examples of each. Bilateral, only one way to get a mirror image halves. Humans, scorpions, dogs. Asymmetry, lack of symmetry, blob-shaped, only phylum peripheral sponges. Radial symmetry, body parts around the central axis, round or circular when viewed from top, only cnidarians or echinoderms. 17. ID the planes. Transverse, sagittal, dorsal, ventral, posterior, caudal, proximal, distal, lateral, medial, anterior, cranial, and frontal. Species. A population of reproductively isolated organisms. Only family members of the same species can reproduce together and produce fertile offspring. Speciation. The development of new species. Evolution leads to new organisms changing over time, from the common ancestral form to new forms that are best suited to living in their particular environments. 19. Isolating mechanisms. Geographical, ecological, temporal, behavioral, and mechanal. 20. The environment selects traits for the organisms with the most advantageous traits in that particular metazoa. A major group of multicellular eukaryotic organisms in the kingdom Animalia. 22. 3 environments where inverts are found. Marine, brackish, simple. Freshwater, more advanced. Terrestrial, land, complex. 23. With most animals, bilateral symmetry is used most often, with that being split into 2 mirror parts. We can see what animals have similar structures in the past. 24. Identify and define these. Subtilization. Concentration of neurons at one end of the animal. Coelom. The fluid-filled cavity between outside of gut and inner body wall. Acoelomate. Having no open body cavity. Pseudocoelomate. False cavity. Tissues derived from mesoderm, only partly lined fluids fill the body cavity of these animals. Coelomate. A group of animals possessing a fluid-filled cavity within the digestive system is suspended. Segmentation. Repetition of body parts, segments. Usually see distinct lines on the body. Planktonic. Carried by sea currents. Sessile. Non-moving. Mesoderm. Muscular system. Reproductive system. Peritoneum. Calcareous. Plate and sea endoskeleton. Ectoderm. Epitherm and nervous system. 25. The multicellular theory. Organisms in a colony specialize to perform specific functions digestive and reproduction and defense. 2. Organisms can no longer live alone and are dependent on zooids in colonies. 3. Colony is now one multicellular organism. Subkingdom protists. 26. All protists share lack of cell wall, at least one motile stage in life. Most ingest their food. 27. Many protozoans are the producers of food webs, accounting for about 70% of all photosynthetic activity on Earth. 28. Types of symbiosis. Parasitic, one species feeds on, but one doesn't immediately kill. Mutualism, interaction where both species benefit. Commensalism, interactions where one species benefits and the other one is unharmed. 29.